Hello everyone, welcome back to RPIIT Academics. In this video, we are going to learn about PAP stain. So PAP stain's full form is Papanicolau staining. So this method is used for routine staining procedure which is used in the cytopathology lab. This technique is named after Dr. George and Papanicolau, the father of exfoliative cytology, and was devised for the optimum visualization of cells exfoliated from epithelial surfaces of the body. The use of the Papanicolau stain results in well-stained nuclear chromatin differential cytoplasmic counter staining and cytoplasmic transparency. Then it is a polychrome staining reaction designed to display the many variations of cellular morphology depending on degree of cellular maturity and metabolic activity. The three main advantages of this staining procedure are: this is a good definition of nuclear staining, it gives very nice cytoplasmic transparency, indication of cellular differentiation of squamous epithelium. Then here is a principle for PAP stain. The classic form of PAP stain involves 5 dyes in 3 solutions. First one, hematoxylin, that is in a violet color, then OG6, which is orange G6 in orange color, then eosin azar 50, that is in light blue. So hematoxylin violet, a basic stain which chemically affinity for acid substances, that is nuclei of cells, which is filled with DNA, that is a deoxyribonucleic acid. Then OG6 orange, an acid stain that reacts with superficial squamous cell, which is filled with keratin. Then there is a eosin azar 50. It is an acid stain that reacts with the cytoplasm of mature squamous exocervical cell, which is basal, parabasal and intermediate cell, and with glandular endocervical cells. Then now here is the diagrams for the stains. So here you can see in the result 1, hematoxylin stain has given the nucleus in a blue color. Then there is a orange stain in a second result, and there is a third stain which is polychromatic stain. So this is how that's why the stain is also known as polychromatic dye. So in the first staining step, the nuclei are stained by a hematoxylin solution. The nuclei are stained blue, dark violet to black. The second staining step is cytoplasmic staining by orange staining solution, especially for demonstration of mature and keratinized cells. The target structures are stained orange in different intensities. In the third staining step, the so-called polychromatic solution is used a mixture of eosin, light green SF, that is and Bismarck brown. The polychromatic solution is used for demonstration of differentiation of squamous cells, for example cervical cancer, and cycle diagnosis for examination under microscope. Then the hematoxylin nuclear stain demonstrates chromatin patterns of normal and abnormal cells. The counter stains orangey, eosin azar have a high alcoholic concentration which provides cytoplasmic transparency. This enables clear visualization through areas of overlapping cells, mucus and debris. Then there is a sample. Specimens can be gynecological smears for PAP stain, and the another name of gynecological smear is a PAP smear. The non-gynecological specimen, which is sputum, brushing, washings, urine, cerebrospinal fluid, abdominal fluid, pleural fluid, synovial fluid, seminal fluid, fine needle aspiration material, tumor touch sample, or other materials containing cells. Then there is a steps of staining procedure. We have 4 steps for the staining procedure. First one, fixation, then nuclear staining, the cytoplasmic staining, and then clearing. So this was it for this video. If you like this video, then please do subscribe our channel and please share it and if you have any doubt, please comment down below. Thank you very much.